Bine v-am găsit, vă mulțumesc foarte frumos pentru invitație, doamna Lăcrămioara Fresineanu, Fundația Dan Voiculescu, Anca Sandu și echipa de organizare. Numele meu este Luisa Baca, sunt medic și cercetător în Anglia și Statele Unite. Am studiat în România. Prezentarea mea va fi în engleză, dar am dorit să vă salut în română inițial. This is just a quick highlight of my journey so far. I'm very grateful for my Romanian roots and the training I have received here, and very grateful for my UK and US training as well, that have brought me to, over the last couple of years, have brought me to learn about geroscience and the geroscience hypothesis, which relates to the fact that aging could be a modifiable process, and that it was also the realization that the root cause of the diseases I was treating in the hospital, for example, heart attacks or atrial fibrillation or other heart diseases, all those diseases come with advancement in age. So, of course, there are risk factors, but aging is the biggest risk factor for all the diseases. So I thought maybe it's useful to prevent and treat before those diseases start. That's my motivation for studying aging. While preparing for this presentation, I was so happy to learn more about the fact that Romania actually is a pioneer in the field. So Ana Aslan was a visionary in 1952 when the Ana Aslan Institute was built and created, and those are in her words, the prevention of aging is the medicine of the future. We are still working in that towards that. I think people don't like so much the idea of prevention or find it boring, but people are currently interested in longevity. So that's a good thing. Just, you know, as background, United States came later than the Romanian efforts, and it was only in 1974 where the National Institute of Aging was created there. So we have good legacy. Both Ana Aslan and Nathan Schock, who's considered the father of geroscience and gerontology, had the same kind of vision and dilemmas in the hospital that any of us have, if you're doctors in the room. But also, if you've ever met, you know, if you have a grandparent or a parent, or, you know, if we look in the mirror, and that's the fact that as we can meet someone who's 40 and they look in a certain way, and we meet someone who's the same age, but they seem to have a different biological age, or they can be more fit or less fit. So how do you differentiate? Because basically, chronological age is quite a boring concept. It flattens out the differences, and in medicine we figure out that, you know, the risk factors are different, even if two people have the same chronological age. So phenotypic age means exactly this difference. Someone in their 70s can be very, very active, and someone of the same age can be less active. And what explains that difference? So as we age, there are factors that, I guess, attack our resilience and our youthful body, and those are, I put them here in red. And also there are factors that want to stabilize and keep us healthy and youthful, and this balance changes over time. So as more time passes, it's harder to maintain the balance. And you can see that when we're babies, we all look the same. As we age, we start to look a bit more different, and at the end, we look very different. So there's a lot of heterogeneity. And I'm very interested because people have different risk factors for diseases. So the diseases I'm trying to treat, I can't understand them purely by looking at the person's genes. I can't understand them purely at looking at their age. So I'm interested in the combination of all these factors. Okay, so That's biological age. You've heard, and thank you very much to Professor Patrick Payne and Professor Obido Gray for talking about hallmarks of aging. So this is currently the state of the art. This is a paper from 2013 and then an updated version in 2023 where scientists have decided that kind of these are the molecular mechanisms behind that heterogeneity and the biological age. So we are able, if you measure this in a young person versus an old person, you'll see more stem cell exhaustion, cellular senescence in the older person, and so on. I don't want to be extremely technical today, but kind of this is what we see different between young and old. Okay, and then technology has evolved since Anna Aslan and Nathan Schock, and now we're able, especially in the last 10, 15 years, to measure so many things in our blood. So those would be here the, oh, I don't have a pointer, but okay, the omics. But you can also measure your heart rate. You can wear a Fitbit, a Whoop, a Garmin. I'm not promoting any, but, or you can measure your weight on a special scale. So you can integrate a lot of data. That's where we are today, and that's what's different from 50 years ago. All right, so what do you do with, this is what we think goes wrong with aging. We can now measure loads of things. All right, so I am interested in how we could make good use of all this data in clinical practice. Can I actually do anything with it? So I'll talk today about the blood tests and the biomarkers of aging in a tube. Okay, so this is a very simple process. You take blood, you send it to the lab. We then investigate markers from these cells in the blood. And there are fancy terms for anyone new in the field. Genomics, which look at the DNA, you heard earlier this morning. Epigenetics, what happens to the genes. Transcriptomics, proteins, and metabolites, which are different from proteins. Okay, cool. So all this data, again, it's not anymore just a couple of blood tests. There's thousands and thousands of elements that you can measure, and you've heard some markers earlier from the other speakers. What people have done have used statistical models to try to make sense of so much data. And Professor Steve Horvath, about 10 years ago, realized that when he looked at the DNA methylation changes, so those were in this domain here, the change in genes, so epigenomics, he realized that you can, with a special algorithm, you can detect from the blood test the chronological age of a person. Okay, that's good, but you can just look, you can ask them how old they are, so that's not a major thing, but it was still a breakthrough. Why would blood tests correlate with one's age, right? So since then, the field of biomarkers has really taken off. And in recent ages, I have been working with an extraordinary group of people. These are some of my collaborators, not all. Some are in the room, some are not. But we have been trying to make sense at the Biomarkers of Aging Consortium and Healthy Longevity Medicine. We have been trying to make sense of what kind of clocks, what kind of algorithms are good, and how can they be used. So, spoiler alert, none of these aging clocks that are on the market, the agreement is that we don't fully understand what they do, but they definitely send us biological signals. And my message today is that investment in research, including in Romania, is really important so that we can make sense and use technology responsible in the benefit of all patients. Okay, so with my colleagues, I'm working on 2 projects. Very briefly, one of them is a conceptual paper where we're discussing if there would be a perfect aging clock, so perfect algorithm based on blood tests, what should we do with it? So that's project 1. And basically, we are saying that it can be integrated potentially with current medical tools. So in clinical practice, you use risk scores. I use risk scores to determine if a patient will have a heart attack or not in 10 years from now. I look at their cholesterol level, I look at their fitness level, measure their ECG and so on. But most importantly, most, if not almost all our risk scores include chronological age in the calculations. So again, these are statistical scores and epidemiological scores. So the idea would be, how would the future look like, where not necessarily replacing from these scores the chronological age, but introducing as well the biological age. So again, if I have a patient that's 70 and another patient that's 70, and they have similar risk factor background, by doing a bit more personalized and further testing, can we detect the risk better and basically personalize their treatment and their care and their prevention regimen. So that's the idea of this first project. Just to say that, clinical decisions are very complicated, as most of us know. One blood test is never going to solve anything, so it's about integrating these tests and any biomarkers into well-defined algorithms where we already have evidence about what works and doesn't work. Okay? One thing we've learned over the last few years is also that not only do we age differently one to another based on what we do, based on our genes, but also our organs age differently. And this is work from the United States, but also other countries in Europe, by Gladyshev Lab and Tony Viscorey. And I'm particularly interested in the cardiovascular system. And just to say, for example, this is a risk score. If I were to have a perfect blood test or a biomarker that tells me better about the age of the heart, I could introduce it in one of these scores. Then the second project, and I'll finish with this. In the second project, we wanted to look at the current evidence that is available. Are any of these aging clocks ready or have enough information to be correlated with any of the things that we are interested in clinical practice? This is a project with Professor Meyer from Singapore, but also the rest of the consortium. And here we found out, this is a preprint, we found out that second-generation clocks, like you've heard also this morning, which predict mortality, have most evidence. It's not about whether they truly show biological age or they're the better, but there's more evidence for these clocks. So if you do have a longevity clinic and you really want to apply an algorithm to a big panel of blood tests, you could say that there's more evidence to apply something like Grimmage or other clocks from this second generation. All right, but again, you can see in red the fourth generation, which actually indicate that if the blood test says causality. So we see changes with age, but we can't demonstrate that what's in the blood test really is the reason why we age and if we age in a particular way. Okay, again, my collaborators, I'll end on this theme and say thank you so much for listening, and thank you so much for being here today. I think Romania has great potential to take on board the ideas here, work towards prevention, and think of aging as a modifiable process. And let's live longer, better. Thank you.